everyone, myself Shakina Khan, and today I'm going to present my literature before you on topic identification of novel blood biomarkers of treatment response in cystic fibrosis. So, giving an introduction about me, I am finally a B.Pharm student from Siagat Institute of Pharmaceutical Science. So, starting my presentation before you all, before first talking about a corresponding author. So, the corresponding author of this research paper is Bradley Quinn from MD UBC Division of Respiratory Medicine, UBC rank. He is currently as an associate professor. His area of focus is Dr. Cohn focuses his research on bridging discoveries in the basic laboratory into the clinic to improve patient outcomes in cystic fibrosis. His major discoveries are like biomarker discovery, biostatistics, cystic fibrosis, and health outcomes. So, moving ahead, the background and significance of study, like my basically why I choose this literature for my presentation is like it has very huge background and it is very significant for nowadays, and it is important to study this. So the basic points are: it gives personalized treatment, monitoring direction progression, early detection, research and development, understanding disease mechanism, and clinical trial designs. So, talking about the first point, that is personalized treatment. So, biomarkers can help tailor treatments to individual patients by predicting that how they will respond to specific therapies, leading to more effective and personalized care. The second point, like monitoring disease progression. So, they provide a way to monitor disease progression and treatment efficacy non-invasively, allowing for timely adjustment for treatment plans. The next thing, that is early detections. So novel biomarkers can potentially identify disease exacerbations or complications early, improving intervention outcomes and patient quality of life. It has also it is also significance in research and development. They aid in the development of new therapies by serving as endpoints in clinical trials, helping to assess the effectiveness of new treatments more accurately. The second thing, which is understanding disease mechanisms, like it is very significant in understanding disease mechanisms. We can make a new discoveries. Discovering a new biomarkers can enhance understanding of CF pathophysiology, potentially uncovering new therapeutic targets and contributing to the development of innovative treatments. So yeah, it is very useful in clinical trials design. In clinical trials, biomarkers can be used as surrogate endpoint to assess the effectiveness to new treatment more quickly. So moving ahead on the introduction of the topic. Yeah. So basically, cystic fibrosis is a life-limiting autosomal recessive disease affecting over 70,000 people worldwide. So pulmonary exacerbations are clinically impactful events for individuals with CF, CF, that is cystic fibrosis. Unfortunately, many CF individuals with pulmonary exacerbation fail to regain their baseline lung function despite treatment. The main objective of this study was to use unbiased proteomic technology to identify novel blood protein biomarkers that changes correlate with clinical response by the end of treatment. So biomarkers that are measured objectively and reproducibly have been studied in order to help guide the therapeutic interventions in diseases such as COPD. So suboptimal pulmonary exacerbations outcomes may be due to delayed recognitions and treatments. Many of the people are suffering because of cystic fibrosis because there is delay in treatment, and the time they recognize that they are suffering from cystic pulmonary fibrosis, they it is like very late, so they are not able to recover their lung functioning. So these biomarkers are an aid in clinical decision making. So because of this biomarker, we can early recognize and we can early do the treatment that change is potentially more informative in assisting treatment decision, as it provides the opportunity for CF physicians to modify treatments earlier than if they had waited for the patient to respond or not clinically, which can take longer in some patients. Using the treatment, IV treatment, which basically is given in pulmonary fibrosis, cystic fibrosis. So it takes a lot of time, and because of that time, the we are not able to regain the lung functioning. So the main objectives of this pulmonary fibrosis is, like in the pulmonary fibrosis means the researchers, the main objectives of that researchers were to use label free liquid chromatography tandem mass spectroscopy to identify novel blood protein biomarkers, area associated with early responses to IV antibiotics, and to determine early changes. Like if we determine the changes earlier, so it will be very useful, and we can easily treat or cure that the lung functioning can be improved and we can cure the disease. So changes correlate with clinical outcomes by the end of IV antibiotic treatments. The treatment of IV antibiotics it takes a lot of time to recover or to treat these patients. So because of these biomarkers, we can early detect the symptoms and we can make new decisions about how, like, we can recure or how we can cure the patients. So basically, the methodology which were used in these treatments were untargeted proteomics profiling of blood proteins, blood samples and clinical outcomes, analysis of candidate blood proteins, protein-protein interaction network and pathway enrichment analysis, and statistical analysis. So basically talking about untargeted, basically talking about untargeted proteomic profiling of blood proteins. So plasma in this basic methodology, plasma samples were analyzed with label-free liquid chromatography tandem mass spectroscopy. In brief, it was done to facilitate the analysis of less abundant plasma proteins. 14 of the most highly abundant proteins were first immunodepleted using the human 14 multiple affinity removal spin cartridge. So in these various proteins were recognized and identified, like which were like alpha 1 antitrypsin, IgA transferrin, haptoglobin, fibrinogen, alpha 2 macroglobulin, alpha 1 acid glycoproteins, and many more proteins which were used on these studies. So the second, like analysis of candidate blood proteins. In this methodology, the researchers have done, like they have chosen five low abundance candidates blood proteins. They have taken the five low abundance candidate blood proteins, including interleukin, IL-6, IL-8, tumor necrosis factor, TNF alpha, and calpoprotection that are beyond the detection limits of LCMS and MS were analyzed in serum samples. In the next, protein-protein interactions, which we all knew very, like we have studied in like sciences and research views, which are protein-protein interactions. There are many types of protein-protein interactions which take place and it alter the effect of the drug. So the differently expressed proteins identified, the different proteins were identified basically and were applied as inputs for plasma protein I network and pathway enrichment analysis. The string database was utilized to assess the protein functional association. The active protein protein interaction were identified based on experimentally determined interactions, and they were studied and analyzed doing this. So next is statistical analysis. The basically the use of statistical analysis was done to give to gain the overview of the study. So statistical analysis was performed using R version 3.5.0, the R Foundation for Statistical Computing, Vienna, Austria. So continuous variables were presented as mean standard deviations. Thus, all standard deviations were recognized. Categorical variables were reported as number with proportions longitudinal changes in the levels of candidate blood proteins. Yeah, in the candidate blood proteins, and changes in clinical outcomes were analyzed due to this. So these were the basic methodologies which were, yeah, these are the basic methodologies which were studied and used to like for the identification of protein biomarker. So next, talking about the results. So the various graphical results were gained and which were totally based on the, yeah, various graphical results were which were totally based on the like how that 25 patients were recovered and how that 25 patients acted on the various IV antibiotics. So the like basically there was a like we have chosen a total of 25 PEX events, that is pulmonary exacerbation events, and from that 22 unique CF subjects were eligible for these studies. Out of 25, only 22 like events were eligible. Clinical characteristics of participating subject at hospital admissions are summarized. Like they were admitted into the hospitals, and the brief overview and brief examinations of those individuals were done. So in brief, mean baseline lung function was 21.5%. So basically this FEV watch like which we can see. So predicted, yeah. So the graph which is like presented before you, it shows that a brief mean baseline lung function was 63. Like the this is the area which the more number of patients were belonging to this 50 to 75 category. So a brief mean baseline lung function was 63.4 predicted, and 60% of CF subjects had moderate to severe obstructions. Mean FEV percent predicted, and CF respiratory system diary chronic systems. So this first graph which predicted that most of the patients were lying in the category from 60 to 75, and the mean number which was calculated was 63%. The modified FUX exacerbation score ranged from 4 to 8. So these are V1, V2, V3, V4. These are the various subcategories which are shown in this first graph. So talking about the full presenting this chart, so it shows various different functions. So this FEV1, which was predicted, is force expiratory volume in 1 second. It is force expiratory volume in 1 second. And this graph, second graph, we show CFRSD and CRISS. It is CF respiratory symptom, dairy chronic respiratory infection symptom score. So these are the two graphs which shows these two different outcomes. In this first graph, which shows force expiratory volume, like when the antibiotic, like IV antibiotic were given this and given to the patients, and it has been recorded. So this forced expiratory volume in 1 second of the all the patients was examined and recorded. So it show various phenomena. So the age, yeah, basically the age of the all patient was among 34 to 50, the age number, and in which the female suspects were 12, and the genotype were 0, and number of subject was 22. Like we have chosen 25 suspects, but only 22 were eligible for those, like for the study. So secondly, talking about the second graph, which shows yeah, just a second. Yeah, which shows the correlation between this CFR SD and CRISS. So basically, this both graph shows that in first graph, the FEV, FE in 1 second was more in the graph fourth, that is V4, and it goes on declining. The like as the days goes on of the patient, it goes on declining. It shows that the condition of the patient becomes severe as the its lungs capacity was, yeah, or we can say its lungs tissues were losing their property, like property, we can't say property, were losing their capacity of like exacerbation. So talking about the second result which we got, yeah, it is the result is about longitudinal changes in candidates LC-MS blood proteins. So these two graphs basically show serum IL-6 levels significantly. So we can see that as the days, the patients which were admitted in the hospital, as the days goes on, it was decreased from hospital admission to each of the following time points where significant changes were not identified between subsequent time points, which is consistent with IL-6. Like this one, this is of V2 to V1 and this is of V4 to V1. In this graph, only two graph suspects can be seen, and the CRP, APOME, these all are proteins and protein changes. So it is a volcano plot demonstrating blood protein measured with LC-MS with statistically significant fold changes from V1 to V2, and second graph is from V4 to V1. The third result, which was it is of protein-protein interaction, that is PPI network, which was done by the researchers. So it is a pathway enrichment analysis by string algorithm. The online string database was applied to identify the protein-protein interaction network using differentially expressed proteins and their most enriched molecular pathways following IV antibiotic treatment. So the online string database was applied to identify the protein-protein interaction network using differentially expressed proteins and their most enriched molecular pathways following IV antibiotic treatment. The protein-protein interaction networks was constructed by the string algorithm after analyzing 47 proteins. Like among all proteins, these 47 proteins were selected for the research works that changed significantly between V1 and V2. Like in this graph, we can see from V1 to V2, and second from V4 to V1. Yeah. And then top enriched geobiological processes terms. 22 of the 47 D proteins from V1 to V2 were involved in the following: immune inflammatory related geobiological process and regulation of activation, regulation acute inflammatory response, immune effector process, and complement activation classical. So basically we can see, yeah, the network from proteins. It is a network of protein from GG25, LBP, EFE-M1, and PTPR3, PVR, PCM, CFL1. The protein network is shown in this graph of the protein-protein interaction network. So moving ahead, talking about novel technique which was used in this research paper was label-free quantitative proteomics. So label-free quantitative proteomics is a powerful tool. It is a very powerful tool for identifying and quantifying proteins and biological samples without the use of chemical labeling. This approach relies on accurate mass measurement and peak intensity to quantify protein abundance, allowing for comprehensive protein binding. So the conclusion which like I felt up from this research paper, this literature study was like by using the label free LCMSMS quantitative proteomics, we identified several blood proteins involved in complement activation and inflammatory immune related pathways that changed in response to IV antibiotic treatment. Yeah, that changed to the IV antibiotic treatment. Early change in IGFR2 correlated with symptom improvement. Validation as an early hmm, with symptoms improvement by the end of treatment and requires further validation as an early marker for symptomatic treatment response in individual with CF. The key learnings which I got was, first, the very first thing which we are the the researchers which found was identification of potential biomarkers. So these studies successfully identified several proteins that showed significant changes in expression between patients with good and poor treatment responses to cystic fibrosis. And basically, this study leads to improved patient care. Patient can be treated more in more improved manner